So thank you very much for that kind introduction, and thank you very much for the invitation. It's wonderful to speak to amongst a diverse group of speakers from many backgrounds. And my job is a clinical geneticist, and most people have no idea what that is, including my kids. And so basically my raison d'etre is to try and solve clinical problems, and the particular clinical problems that I solve is trying to make a diagnosis, most usually. And most usually that's in children or young adults, often with combinations of problems where it really has been a diagnostic odyssey or a puzzle. So my role is to help to try and solve those puzzles. I guess I'm nowhere near as good as I would like to be, and that's what drives me to look for solutions to the problems. And we're very fortunate in Western Australia to have a number of aligned initiatives, both within our health department and outside, that really help to translate new knowledge towards these problems. So what I'm going to talk about today is some of those, and I apologise to some of you who I can recognise in the audience who've seen some of this before. There'll be some new stuff and some old stuff, and I hope I don't send you to sleep. Why do I do what I do? Well, I think I've kind of just outlined it, but we might take a different frame of reference and say, well, particularly I'm interested in rare diseases, which are individually that rare, but cumulatively they're common, estimated to affect up to 1 in 12 individuals. So then we're talking about similar numbers of people to those affected by diabetes. For instance, that would include upwards of 1.2 million Australians. And I guess coming from initially being a paediatrician, there's no secret that my heart lies in paediatrics, and that might equate to 400,000 or more children. That's 40,000 or more children in WA. These are conditions that are diverse, often affect multiple organ systems, can affect any organ system, and are cumulatively common. These are some stats just outlining some of those. What is a rare disease? There are different definitions around the world, but the sort of one that Australia is sort of adopting is something that's chronically debilitating, is complex and requires combined efforts to treat them, and has an estimated prevalence of 5 in 10,000 or 1 in 2,000 or similarly low. We talked about collectively being common. We talked about some comparators. We can talk about some things that are included in rare diseases, by which we would be including all childhood cancers, a quarter of adult cancer, and they're very significant personal costs, familial costs, and public health system costs. So there's no surprise then that governments around the world have become engaged in this, and there are a number of global efforts trying to harmonise those approaches. And just another number, because people like numbers and comparators, if we stuck everyone who had a rare disease in a country, it would be the 3rd most populous country in the world. The impact is great in terms of they often appear early in life, they can be associated with significant disability and premature death. There's no secret that rare diseases provide us a real window. There's sort of the clarity and the extremity of rare disease. Often the signature in rare disease is so high that it outwashes the noise. And this was really recognised back in the 17th century by Harvey, and I'm not going to read out that quote. I think you'll read it a lot quicker than I can talk. But why now? Well, there's a convergence of technologies, and there's an opportunity to align those to an unmet clinical need. If we can think about global initiatives, and we can think about global initiatives where Western Australia and Australia is a player, then we can think about the International Rare Disease Research Consortium, which is really, it has its 2 main current aims of developing diagnostic tests for all rare diseases and 200 new rare disease therapies by 2020, and you can see that it's represented by the West Australian Department of Health, which is one of the founding members. The challenge really is that there are in fact many different tools and many different solutions, and these are actually, a number of them already existing, and it's about aligning the many facets of the excellent work that's going on currently to that problem. So there's some sort of cohesion around that, some sort of ecosystem, some sort of knowledge management system that can really drive the solutions from the perspective of patient need and feeding back to the patient. That all sounds wonderful, doesn't it? How are we going to do that? I might just talk about a couple of aspects of that, because there are many issues. But you would have already guessed that diagnosis is kind of one of my passions, because that's the entry point. If we don't have an accurate diagnosis for a patient, how can we treat them appropriately or prevent treating them inappropriately? How can we understand their disease? How can we track their disease? How can we understand how their disease evolves? How can we understand how this person with a particular disease is going compared to another person with the same disease? Because that's really important information for managing a patient. That all starts with getting the diagnosis right in the first place. And we know it's particularly challenging for rare diseases, for instance, from the European Union, even looking at relatively common rare diseases. So the sorts of rare diseases that paediatricians and physicians should know something about. That a quarter of those individuals waited from 5 to 30 years to get a diagnosis. And in 40% of the time, the first diagnosis was wrong. So there have been some really fabulous advances in trying to make diagnosis in rare disease, and I'm sure a lot of you would be swept up in the wave of genomics and next generation sequencing and some other terms, or massively parallel sequencing, and really that they are making step changes in how we go about making diagnosis. They generate, I'm not going to say big data, they generate significant amounts of data, and really what we need is quality data, because like anything, it really is garbage in, garbage out, and I'll illustrate some of those points. It's particularly a challenge in rare disease, and why so? Because the diseases are individually rare, and there may have been siloed efforts to collect this information. Getting it in a cumulative source is a challenge, but the one that is being addressed. And equally, because of the way this has evolved over the time, some of the information is plainly wrong. So if you go to public databases and look, say, is this variation I found in a gene actually going to stop how this gene works and cause a disease? And go to this database all about this gene and look, it says it does. Well, actually, about 1 in 8 times that's wrong. And there's some reasons why there's some good reasons why that's wrong, and we really want to reduce that sort of error rate. We need, in order to do so, we need to get data in the first place. And as clinicians, we're lazy and we don't want to do anything differently to what we already do. And so really we need to align those ways of capturing data to how clinicians work, to their workflow. It has to be at least time neutral and give us added value. Otherwise, there's a real cost otherwise. If we're doing extra data entry, extra data collection, and actually getting no greater output, we're actually going to get less output, and it's actually going to cause harm for patients. We need standards and we need objective measures. There are some excellent tools really aimed at mimicking clinical workflow to enter that data. This is one developed by some colleagues at the Sick Kids in Toronto, which we're fortunate to collaborate with. But anyway, it can be configured to your workflow with automated measurements and those sorts of nice built things, and some inbuilt diagnostics. We can capture information in different ways and more objective ways, and one of my research interests is 3D facial analysis. So in this case, it's using 3D photography essentially to take, we take 10,000 data points, you could take many more, and we capture it in less than 2 milliseconds, and then we use that information to try and create diagnostic signatures. And I'm not going to talk much about that today, but I'll come back to that. We've got this, okay, now, so we have this wonderful world, we've got all these technologies and we've collected all this data, now we need to integrate this data so we can apply it to this problem, which is trying to get a diagnosis early, so we can give patients their best management and inform families in the best way. And so really we need a structure around that data. I'm sure a lot of you in this audience are experts in ontologies. I'm not. I have a limited understanding of ontologies, but really ontologies in the philosophical sense is all about meaning and structure and how things kind of relate and who am I and where do I fit. And then there was a movement through sort of AI and then through to biomedical research. In medicine, what does a doctor usually use to try and make a diagnosis? Well, it's really the manifestations of that condition. What signs and symptoms have you got? Those are the clues we use to make a diagnosis. That's a phenotype in medicine. So a phenotype ontology is really just a language, a way of talking about, a way of structuring, a way of making interoperable that language. So then we can get computers to help us reason, to help us out, basically. So then we can create those structures and those relationships, and then we can apply reasoning against those to try and make a diagnosis. So a doctor out in Joe Blogg's land, who has never seen said rare condition, types in a list of symptoms, and on the back end of that, you could have a diagnostic that says, well, have you thought about this? You've probably seen this, but this might be the diagnosis. So it's a way of trying to upskill people and really to enable non-experts and experts. We need to combine the data. So we can think about a number of ways of doing that. If we think about doing that in terms of aggregating the data, there are many ways to do this. This is Professor Andreas Zankel from the University of New South Wales, a particularly clever and extremely lovely man who together with Tudor Grozer and others, developed a really beautiful tool called Skeletome, which aggregates data initially in one domain, which is just for bone problems, and subsequently has been generalised out for other domains. And really it was about capturing all that knowledge that exists in radiographs, that exists in gene tests, that exists in clinicians' descriptions, and equally with a view to collecting how patients describe their own conditions, because that's actually really powerful information for diagnosis, and then creating a structure around that to enable diagnostics. We can also look at different species, but we can only do that if we have the same sorts of structures for those other species, and we actually do, which is pretty cool. So we can actually look at how things vary between and are similar between different species to give us clues about things. You know, see kind of ontology kind of written through this. I love this, the International Mouse Phenotyping Consortium. So this is a group of a worldwide consortium led out of the UK, where they're basically looking for manifestations of ill health and disease in mice. And some of the ones that I really love, it probably doesn't project very well, but one of these is the acoustic startle, which to me is some guy in a white coat going boo behind a mouse and seeing what happens. But actually there's very powerful data available from mice to actually help interpret human data. We know this historically. We also know this from gene technology. So we know this, that when we use these new powerful gene technologies, we get a lot of data and it can be really tricky to interpret. And if we just try and interpret it on frequency data, chemical variations, biological data, yeah, that gives us some good sieves. But it's not perfect. But if we then marry that with other data, how do diseases manifest in humans and, in fact, in other species, then we actually get step changes in the usability of that data. So this is one example of that, which is a collaboration between the Sanger Institute, a very famous institute in the UK, and the Charité University in Berlin, who would be equal kind of standing in German genetic eyes. If we just filtered information just on how frequent were these things that we found when we did these powerful gene tests, we get a certain level of information and a certain ability to actually resolve a problem. If we didn't actually do the gene test at all, and we just looked at the manifestations of the disease and how they cluster and how they relate, actually it's more powerful. But of course, it's when you combine the two that you get the real step change in the ability to make a diagnosis. So it really talks about the power of mouse and a man, and I think we know how very powerful this mouse and this man was. And when we're talking about facial information, well, we've got to understand how variation is in the normal envelope in a sort of a multi-dimensional space compared to how it might play out in a disease state. We're very fortunate in Western Australia to have a normal range of high-dimensional facial variation with about 1,000 individuals, predominantly young individuals and children, to base these analyses on, if you like, and we can do silly things like create synthetic faces that you can see really couldn't exist. But you can do more interesting things. You might move through this data in different ways. For instance, you can move through on whatever particular metadata you collected to see how it varies on age, how it varies on gender, body mass index, a particular disease state. With some work with some colleagues in Belgium and in the States, we wanted to take another approach and say, well, can we take a 3D facial photograph and actually turn that into some sort of text-based description? Because we do have, believe it or not, standard descriptors for facial differences and how they relate to certain diseases. So we wanted to actually unlock all that text-based information. So how do we go about that? We basically code the face to text. So that's essentially what we did, which really allows us then to back it onto these ontologies, how do all these things relate, to get another way of trying to enable a diagnosis, or just to really even just use it to generate a medical report, or as a learning tool. So we get to this point where we can say, well, our faces actually can tell us something about our DNA. I'll show you a couple of things a little bit later. Can our DNA tell us about our faces? This paper got a lot of hype. Which is kind of good and bad, because this was really sort of looking at forensic sort of applications, and it did get a lot of hype. Bottom line is, DNA to face is a long way away, and it'll probably never happen, other than a crude representation of a probabilistic range of a face which is very wide. That is because of a whole different bunch of reasons. But basically, the path between gene sequence and manifestations is a very long one and a very convoluted one with lots of players. So just looking at gene sequence and saying, what is your face going to look like? Not great. But there was some level of capability if you looked at lots and lots of individuals and giving an average kind of thing. So it's not like you're going to get a DNA sample and be able to say, that's your perpetrator of your, so you can generate their face, which is, of course, the angle the press took. You can aggregate this data at an international level, so particularly in rare diseases, we need to do this because they're individually rare. So this is an NIH-based collaborative, which we're fortunate to be the Australian node for, where we're collecting this information to develop new diagnostics in an international sense. We can collect other sorts of information. So this is BioLark. So some of the people we talked about with Skeleton are in fact some of the principal architects behind BioLark. And BioLark is a way of, I like this term, it's biomedical large knowledge collider. Basically, it's dragging a whole bunch of information and using ontologies and trying to answer questions. And so some of the applications behind that are being now applied to things like mining letters that are written in free text to try and enable diagnostics. We can combine patients. So in rare diseases, it's very powerful if we have a few individuals with the same rare disease. It's powerful in trying to find the cause of their disease if it's not known, for instance. But actually trying to identify those individuals can be really tricky, particularly when you don't even really know what disease they actually have. So now there are international matchmaking initiatives that basically take packets of genetic information, packets of the phenotype, how does the disease manifest, and then they match them and say, well, hang on, these 2 patients or these 8 patients that have been entered from anywhere around the world actually are really similar. Now let's go back to their genetic information, and those couple of things that you found in those 2 cases that you couldn't call by yourself, wasn't enough information, actually they both happen in this set of patients. It's looking like that's the cause. So now we can call that. We couldn't call it before. Why do I get so interested? Again, it's just, to some degree, I'm a technology, can I use the word slut in the audience? In that, I don't really care. What I want to do is apply something to a problem. And there are many overlaps in analytics, and I can see some of our Deloitte representatives. Yes, I'm looking at you in the audience, and this is we talked to the guy who was doing multispectral imaging from planes to map fire ants. I think was the application. But you can think about the difference between surface analysis of a geographic area. You can think about surface analysis of a face and what sort of analytic overlaps there might be there, what sort of joint learnings there might be, how we might apply those technologies from one domain to another. Or if you talk to one of my friends who runs a petroleum consulting company, he'll show you some outputs of his surface analysis. This is how he designs where to put his wells. He gets a whole bunch of information from the surface, and then he uses that surface information to say, I predict we need to put a well there. That's essentially a diagnostic. And it kind of looks a little bit like a face if I pick the right image and turn it on its side. So really, data analytics, what's that to me? That's a method to try and help solve problems for the patients that I see. Be that at the level of a facial analysis, which really is increasingly moving into the clinical realm of translating, but I would say is still primary research application, whether it be a genetic diagnostic or the combination of the two. I'll just quickly run you through a couple of these. So we have different ways of representing the data, and basically what we do is we create what we call normal equivalence. So we create essentially what that individual would be without the variation that's outside some sort of predefined limit. Okay? And we do that in a way that looks at the whole face. And then we can do that for individuals or groups of individuals. We can create signatures in terms of heat maps of where those differences are and where they go. So this is a where they go, a vector map. And so actually at the ends of all these arrows is where a group of individuals with this particular condition would be. And this is a 3D space. So if you flip this round, you can see those arrows actually going in behind the cheek. Anyway, what that computer spits out actually fits with the clinical description of the illness. And that's pretty cool, because these are biologically dumb algorithms. They don't know biology, they don't know medicine, they're just interpreting a set of numbers. So when they actually came out with the stuff that we clinically expect, that was kind of neat. Equally, we can apply these technologies for monitoring disorders. You would think that's actually straightforward, but actually in medicine that's a huge challenge, actually having effective monitors to tell you, is your drug working or not? So for a particular rare condition where there's a problem in body metabolism, where things basically build up in organs and the patient dies, usually as a young child, then there is a particular couple of treatments where you basically give them back the stuff they're missing. And when I'd watch this happen, both in terms of giving it as a medicine, or if you give it, actually you can transplant individuals, because you actually give them new white blood cells which make the enzyme. So transplantation is in fact the first genetic therapy. I've watched in time in clinic when I was working in paediatric oncology how the faces of these individuals would change. So we thought, well, why can't we monitor this with 3D facial analysis? So we did. And you can see over time with treatment how those differences in the face are slowly resolving. Or if you like numbers, we can put numbers. If you want graphs, we can generate graphs. But I like pictures. This is what we need. What are these? Yeah, so we need T's, don't we? We need time, treasure and talent. So any smart people out there who think they can help us, please come and speak to me. And if this will work. Always worrying, putting a bit of a gimmick in your presentation, isn't it? Thank you. Yeah.
Really interesting presentation. Have you heard much about like Watson parts and
yep.
What do you think of that, and do you see that as a great?
Do you mean Watson parts or IBM's Watson IBM's Watson program? Yeah. So we've got some colleagues that are using Watson for those sorts of reasoning applications. So what I think of it, I think it's great.
And you guys have, your semantic mapping and all the autosomal stuff. You guys, is that publicly available information that you work and push?
Right. So in terms of the ontologies. The ontologies are publicly available and evolving, and there are ontology warehouses that could tell this information. The one that we use the most in the rare disease space is called the Human Phenotype Ontology. The Human Phenotype Ontology.
Again, very, very interesting presentation. Do you believe that your data sharing, say for instance the data you gather from, say for instance, facial mapping, creating linkages will allow you to more effectively help you elucidate a model?
Absolutely.
So,
absolutely.
Is that type of work being done?
Yeah. So the NIH US National Institute of Health-based initiative is for faces is exactly that. It's creating a centralised repository to deposit information to develop better models.
So obviously, the more data sets that you have,
and the more variation in your data set. So if you send me 5,000 of exactly the same thing. But the more variation, and that's it. And what that actually means at a global level is you actually need really good representation of different populations around the world, because that's when, if you understand all those different levels of variation, that's when things really sink.
How do you actually remove the noise that's created through normal normal phenotypic development?
Yeah. So the way that we approach is because what people used to do, and still do sometimes, is create averages, and they say, here's a set of individuals with a disease. And this is the kind of the average kind of face that they got. And here's that kind of average normal kind of face. But then there's this big error on this. So we took a different approach and said, well, actually, we want to look at how all the individual facial points, those 10,000 points, relate to each other. What's the harmonious relationship between those? And then we'll define that. Okay? And then we'll compare that to how that happens in a disease state. It's a very different sort of comparison, which it does exactly what we're talking about. It's trying to remove that noise and that overlap.
I'm just glad that you moved on to actually having a solution for something rather than a problem that you can actually solve by using data.
I'll give you lots more problems.
It's fascinating.
Okay, thank you.